Hello students, today we are going to study structure of DNA. DNA is the largest macromolecule and this is the genetic material in all organisms except some viruses called retroviruses. As you can see in the figure, DNA is a double helical structure. In this helix, steps are formed of base pairs and backbone of the helix is formed of sugar and phosphate groups. Now if we see pitch of this helix, this pitch complete turn of helix is of 3.4 nanometer and one complete turn of this helix is consisting of 10 base pairs. Hence we can say the distance between two consecutive bases is 0.34 nanometer. Now if we observe diameter of this helix, diameter is 2 nanometer. In this helix, two grooves are formed. This small groove is known as minor groove and this big groove is known as major groove. DNA is called a polynucleotide. That means it is made up of large number of nucleotides. If we observe this figure, in this figure, this is one nucleotide, this is second nucleotide, this is third nucleotide, and this is fourth nucleotide. That means this is a tetranucleotide. A nucleotide is consisting of three components. One component is a phosphate group. Second component is a pentose sugar. Pentose sugar means a sugar made up of 5 carbon atoms. This sugar is deoxyribose in case of DNA and ribose sugar in case of RNA. Third component of DNA is known as nitrogenous base. In DNA, there are two types of nitrogenous bases, purines and pyrimidines. Purines are of two types, adenine and guanine, whereas pyrimidines are of three types, thymine, cytosine and uracil. The basic difference between purines and pyrimidines is that of rings. Purines have two rings, one hexagonal and one pentagonal, whereas pyrimidines have only one hexagonal ring. There is a pentose sugar in nucleic acids. This pentose sugar is ribose in case of RNA. This ribose sugar has one 2'OH group, whereas deoxyribose sugar in DNA does not have this 2'OH group. Two chains are held together by hydrogen bonds. Adenine, a purine, is paired with thymine, a pyrimidine. Similarly, cytosine, which is a pyrimidine, is paired with guanine, a purine. Pairing between a purine and a pyrimidine ensures constant diameter throughout the helix. We can see there are 3 hydrogen bonds between guanine and cytosine and 2 hydrogen bonds between thymine and adenine. That means GC bonding is stronger than AT bonding. Two strands of DNA are called antiparallel. That means they run parallel but in opposite directions. If we observe this strand, this end is called 5' end and the other end is called 3' end. In case of other strand, this end is known as 3' end and this is called 5' end. These two strands are running in opposite direction. That is why two strands are antiparallel. This end is called 5' end because phosphate group present on fifth carbon atom of sugar is not involved in bond formation, and this end is called 3' end because OH present on third carbon atom is free. Similarly, this OH present on third carbon atom is free. This end is called 3' end, and phosphate group present on fifth carbon atom is free. This end is called 5' end. Hence DNA is antiparallel. Thank you so much, students. I hope DNA structure is now clear to you.